If you're seeing this video, you've requested information on disease resistance in plants. And the system that I'm going to be describing is one that was given to us by an Australian scientist. I.A.M. Cruikshank was his name. And it has, what we'll be covering today has to do with plant disease resistance, and the plant that we selected is a pea plant. This is the item that handles all of our research, this little pea pod. And it's split open, and we put our treatments on the inside, and then we follow it. The whole disease resistance reaction occurs, the successful one occurs within 6 hours. The type of resistance that we're going to look at is found in a lot of plants. In other words, plants resist most of the microorganisms that are out there in nature. Only a few of the microbes have figured out how to be a pathogen of peas or a pathogen of beans. And the system that we're using is we take a bean pathogen, we put it on the pea pod tissue, and that is a resistance reaction that we see. And the pea is able to totally resist the bean pathogen. However, we also have data on what the pea pathogen does, and we'll try and incorporate into that what the susceptible reaction looks like. So what we have here is a scheme. We have a bean pathogen on pea cells, and the bean pathogen has two nuclei, or well, more than one nuclei, and the host has a nucleus. So we will be following the treatments coming in from the bean pathogen into the pea cell, and this is a scheme that we're going to be looking at right here. This top part is the bean pathogen, and it has products which it can use for signaling. And the two signals that we're most interested in my lab is for chitosan and DNase. Chitosan comes from chitin. The DNase comes from what this fungal cell needs to survive. Those two signals, can you still see me okay? These two signals come into the plant nucleus, and the plant nucleus has a DNA molecule inside it. This is a meter stick that's 1 meter long, and in nature all of this long strip is condensed into a little area that is 10 microns. So it's such a small container that you have to have a microscope to see it. So that's what we're dealing with. And we found in the years of research in my lab, incidentally I'll say what my lab is. I'm a Professor Emeritus for Plant Pathology, Washington State University. And most of the data you see here was accumulated by me in cooperation with a lot of very good students. And if you're wanting to pick this up, you can Google it under my name and just say Hadwiger LA and then non-host disease resistance, and it'll pull up all the publications. So there's quite a few of them, and you'll get bored reading them, but we're picking out the vital parts of what I'm going to show you today. Okay, there's the bean pathogen, like I said, and it gives off chitosan, it gives off DNase. It goes to the nucleus, and the DNase is able to make little clips in the DNA molecule. The chitosan is based up of a polymer of glucosamine molecules, and they have a strong attraction to the positive charges of the DNA in the plant nucleus. And when that happens, this wadded up DNA, which is covered by a lot of proteins and whatever in the nucleus, starts to become loosening up. And some of the genes which were silent before become active. And as years went by, we found there's more and more genes being active, but there's up to 500 genes that become active, but there's only a small group of genes which are vital in terms of disease resistance, and those are called PR genes. And just to pick out a couple of those, that defensin is one, and as a pathway in the plant that produces phytolexin, that's the other. But there's a lot of other proteins that are produced that have some negative effect on the fungus. One of the genes that was already in the plant before the induction occurs is the gene for producing chitinase. Chitinase is an enzyme which, when it touches the fungal cell wall here, which is full of chitin and chitosan, it starts breaking things down to cell debris. And that's where you come up with the chitosan that we talked about. The DNase is just a natural component of the fungal cell. And we found it released by quite a few different fungi. In fact, one of the cures for bladder cancer is put in attenuated TB cells, tuberculosis cells. The tuberculosis cells also release DNase, and when they go into the bladder, they induce an immune response. So the immune response is not necessarily relegated only to the plant. Okay, so the other thing that occurs when all of these responses start developing here is that the fungus growth is slowed down all within the 6-hour period. Now, if you're growing the fungus independently of the plant, there would be a growing tip that have live nuclei in it, and then they're back in the back of the old mycelia. You'd have hydrolytic enzymes which are breaking down components of its own self and sending them up to be incorporated into the new growing tip. But when you slow that down, these nuclei, or these hydrolytic enzymes, which are back here, start easing up and showing up in the growing tip. So by slowing the fungus down, you cause damage in the fungal nuclei. And that damage is enough to keep the cells from dividing. If you don't have cells dividing here in the tip, you will not have any growth. The true pathogen has somehow been able to preserve some of its nuclei here, and after this surge of resistance kind of tapers off, that fungus starts growing and it grows all over the plant, and you have a susceptible reaction. So a lot of the same things happen in the resistant reaction and the susceptible reaction. Is that the difference, the big difference, and this is not determined by me, by a guy named Al Bell really early in the field. The big difference between resistance and susceptibility is the speed of the response. So in a resistance reaction, everything occurs very quickly, within a couple hours, and the fungus stops growing. In the susceptible reaction, it grows a little too slowly, and even though some of these hydrolytic enzymes that get to the tip, it doesn't stop them completely. So that's the difference. Speed is the difference. There's a lot of ways in which these two components can work, and they're both DNA specific. These are the things that activate the defense response. And one of the ways that chitosan does it, and incidentally, chitosan itself is antifungal, so it's also a resistance reaction. I'd like to say a bit about what the function of the compound chitosan is. Chitosan is very closely related to chitin, except it's made up of glucosamine molecules instead of N-acetylglucosamine molecules. That little extra part of the sugar molecule is detrimental to attaching to the DNA. But chitosan does attach to the DNA, and when it does attach to the DNA, it can replace proteins that are holding some of the DNA together. It can also alter the DNA conformation. So DNase, as we mentioned before, doesn't slightly different. It makes clips in the DNA molecule. And when you do the DNA molecule, you have that whole 1 meter of DNA that's wound up in a little tight ball become loose. In both cases, a ability of the plant to transcribe some genes that were silent improves by putting pressure on that little wad of DNA within the nucleus. After those changes occur, the new genes are produced, primarily what they call PR genes, and pathogenesis related is what the PR means. But I'm only going to talk about one. And one of the proteins produced, it's actually a peptide. It's called defensin. And when we found it, we didn't realize that defensin is already a component of many other biological organisms, like the skins of frogs, or scorpion toxin, or even some of our red blood cells have this protein defensin in it. And it is antifungal. So that's one of the main components of the disease resistance. Again, the defensin and these other genes slow the growth of the pathogen. The pathogen has, on a normal basis, we'd have hydrolytic enzymes back here to break down old components of the mycelium, take it up for new growth. But when you slow the growth down, the hydrolytic enzymes, which are back here, ease up and eventually get to where the nuclei, the growing tip, is where they really need the cell division to occur. And when you break up the nucleus, cell division doesn't occur and growth stops. So I'm just going to review all of this again on this bigger chart, and we'll follow it one by one. Okay, the bean pathogen here, the pea pathogen here. The two components which are very able to induce a defense response in the plant nucleus go into the plant nucleus, and we know that they do. And as a result of changes in the plant nucleus, PR products are formed. Some of the PR products are like defensin, go back and they get into the fungus, slowing down the growth of the fungus. And like I mentioned, the normal thing for the funguses to do is take hydrolytic enzymes, break down old DNA and RNA and whatever, and the bases go back up and form the substrates for new nuclei to be formed. But when you slow down the growth, these hydrolytic enzymes come up and they get into the nucleus and they break down the nucleus. And that is really what stops the growth. So that is essentially the basis of my report. I do want to say one more thing about gene activation. Most every molecular biology is looking at gene activation. And there's other ways to do it other than the ones I pointed out here. But there are other ways to change the things that we're talking about in terms of the nucleus, like quinacrine, which is an antimalarial compound, will change the conformation of the DNA. There's other hydrolytic enzymes which will do things to the plant DNA. In total, we've looked at over 200 DNA-specific compounds, and all of them are able to induce these defense responses. So the biology of the pea cell is not much different than the biology of animal cells in terms like the defensin is found in the animal's system, and some of the regulatory proteins that hold the nucleus together, like HMG-A, is found in pea tissue. And when there's less HMG-A, there's less control over the structure of the plant nucleus. So that sums the essence of what I was wanting to talk to you about. We have one small view of how disease resistance works, and it's been done by some very good students in my lab for a long time. And that's the sum of our presentation.